This is your first lecture on the central nervous system. The central nervous system consists of the brain and spinal cord. It was evolutionarily formed by a process called cephalization, which is the development of the anterior portion of the central nervous system. It results in an increased number of neurons in the head, and the highest level of cephalization is reached in the human brain. Here you see a diagram of the embryonic development of the human brain. Essentially, this development starts out as a simple structure, the neural tube, which develops swellings, which are called the primary brain vesicles. You see more complexity developed later on in development, forming the secondary brain vesicles, and each of these brain vesicles does result in adult brain structures. For instance, the telencephalon results in the cerebrum, which is the cerebral hemispheres, cortex, white matter, and basal nuclei, along with fluid-filled structures called the lateral ventricles. We'll go into these in more detail later on. For now, what I want you to remember is simply that secondary brain vesicles result in adult brain structures and also fluid-filled portions called neural canal regions. In embryonic development, the brain grows faster than the skull does. As a result, it folds to occupy available space. A couple of things that happen are that the forebrain gets moved towards the brainstem, which we'll discuss in more detail in the second lecture, and the cerebral hemispheres double back. They cover the diencephalon in the midbrain, while they crease and fold to increase surface area. When we see a picture of the diencephalon and midbrain covered by the cerebral hemispheres, this will be a little bit more clear. There are four major adult brain regions: the cerebral hemispheres, which we'll cover in this lecture, the diencephalon, the brainstem, and the cerebellum, all three of which will be covered in the second lecture. Here you see the adult pattern of structures and convolutions. This is a cerebral hemisphere. It's the largest part of the brain, and it's the portion that's in sort of this peach color. It was also the largest portion of the brain which you saw in your dissection in lab. The area in purple here is the diencephalon. We'll go into more detail about that, as I said, in the second lecture. This area is the cerebellum, which again I'm hoping you recall from your lab dissection. And this area is the brainstem, which is composed of the midbrain, the pons, and the medulla oblongata. In the brain, white matter and gray matter show a similar pattern. There are additional areas of gray matter in the brain which we'll cover, but for now, cerebral hemispheres and the cerebellum have an outer gray matter area called the cortex. So cortex is a general all-purpose name for outer layer. And so this is a superficial layer which covers the cerebral hemispheres and the cerebellum. The cortex disappears in the brainstem, and the gray matter is basically scattered amid the white matter. In the spinal cord, there's a central cavity surrounded by gray matter. External to this, you'll see white matter, which is composed of myelinated tracts. So in the peripheral nervous system, bundles of axons are called nerves. In the central nervous system, they are called tracts. So basically, tracts are essentially the same thing as nerves, except they're located in the central nervous system. They're all about communication. This is a diagram of what I was just referring to. So you can see in the cerebellum, as with the cerebrum, you see a cortex of gray matter. You also see an inner layer of gray matter which surrounds a fluid-filled cavity. In between these layers, you see the outer white matter layer, and you can see that there are scattered bundles of gray matter, which are going to be called nuclei, that are present in both the cerebellum and in the cerebrum. As you move down inferiorly to the brainstem, you see the cortex of gray matter disappear. So you still have inner gray matter surrounding a fluid-filled area, but that's surrounded by outer white matter, again with smaller portions of gray matter scattered within the white matter. By the time you get down to the spinal cord, the only thing that's left is a central cavity filled with cerebrospinal fluid, surrounded by inner gray matter, surrounded by outer white matter. The ventricles of the brain are fluid-filled spaces. You may mistake these in the next slide for structures. I don't want you to do that. They are cavities that are filled with cerebrospinal fluid. They're lined by ependymal cells, and that is no surprise because the ependymal cells produce cerebrospinal fluid by filtering blood plasma. Each of these ventricles are connected to one another and to the central canal of the spinal cord, and we'll see those connections on the next slide. Here you see the lateral ventricles. There's one ventricle in each hemisphere, and they are the largest ventricles in the brain. The lateral ventricles are connected by the interventricular foramen to the third ventricle. The third ventricle uses the cerebral aqueduct to connect with the fourth ventricle. Inferior to the fourth ventricle, you see it narrow down to become the central canal of the spinal cord. The cerebral hemispheres do have surface markings. You see gyri superficially, which are ridges, shallow grooves, which are called sulci, and as you can see from the diagram, gyrus is the singular for gyri, sulcus is the singular for sulci. You also see deeper grooves, which are called fissures, and as mentioned before, there's a superficial layer of cortex or gray matter, and a deeper layer of white matter, which is myelinated. In this diagram, the longitudinal fissure is not pictured. The reason why it's not pictured is because it separates the two hemispheres. You're looking at a lateral view of the brain, so in this case the longitudinal fissure is hidden. If we had a frontal view of the brain, that's where you would see the longitudinal fissure, which is a deep division between the two hemispheres. This is the transverse cerebral fissure. It separates the cerebellum and cerebrum. This is a central sulcus, which separates the precentral gyrus of the frontal lobe and the postcentral gyrus of the parietal lobe. The parieto-occipital sulcus separates the occipital and parietal lobes, and the lateral sulcus outlines the temporal lobes. There are 5 lobes in the cerebrum. The frontal, the parietal, the temporal, and the occipital are all named after the bones which overlie them. The insula is deep to both the frontal and the temporal lobes, as we'll see in just a moment. Here you see another lateral view of the brain. This is the frontal lobe, this is the parietal lobe, this is the occipital lobe, and this is the temporal lobe. If you pull apart sections of the frontal lobe and the temporal lobe, you can see the gyri of the insula deep to them. There are 3 basic regions in all of these lobes. There's a cerebral cortex of gray matter superficially, white matter internally, and deep within the white matter you can see basal nuclei, which again we'll see later on in the PowerPoint. The cerebral cortex itself is a thin, superficial layer of gray matter. It makes up about 40% of the mass of the brain, and this is the site of just about everything that makes you a person. Awareness, sensory perception, the initiation of motor activity, in other words, movement, communication, memory storage, and understanding. Because this layer is really thin, and because it's the outermost layer, you might say that each of you and I have a superficial personality. That's the last bad joke I'll make for a while, I promise. There are 3 types of functional areas in the cerebral cortex. First, there are motor areas which control voluntary movements. There are sensory areas which give us conscious awareness of sensation. And there are association areas which bring together diverse information. For many of the sensory areas have association areas which allow us to interpret the data that are coming in. Each hemisphere is only concerned with the contralateral side of the body. In other words, the opposite side of the body. That's what contralateral means. So the left hemisphere is concerned with the right side of the body. The right hemisphere is concerned with the left side of the body. So if you're right-handed, it means that your left brain is dominant because the left hemisphere is concerned with the right side of the body. There is lateralization of cortical function in the hemispheres, and what that means is that there are some functions that are only present in one hemisphere. In other words, they are lateralized. Conscious behavior involves integration of the entire cortex in some fashion. The motor areas of the cerebral cortex are located in the frontal lobe, and again, these are what control voluntary movement. The primary or somatic motor cortex is located in the precentral gyrus. The premotor cortex is anterior to this. The Broca's area is anterior to the inferior premotor area, and the frontal eye field is within and anterior to the premotor cortex. It is superior to Broca's area, and you'll see all of these in just a moment. So, of the motor areas, here you see the primary motor cortex, and again, please note that this is located in the precentral gyrus bordering the central sulcus. Anterior to this, you see the premotor cortex. Anterior to this, you see the frontal eye field. And inferior and somewhat anterior to the frontal eye field, you see Broca's area, which is the area that is outlined by dashes here. This is simply a mid-sagittal section of the brain that shows you the frontal eye field, the premotor cortex, and the primary motor cortex. The primary motor cortex consists of the large pyramidal cells of the precentral gyri. These cells have long axons that leave the brain through the pyramidal tracts of the spinal cord. What the primary motor cortex does is allows us conscious control of skeletal muscle movements. And the areas of the primary motor cortex can be mapped by what are called motor homunculi, which we'll see on the next page. Basically, these are caricatures that represent the contralateral motor innervation of body regions. This is one way that a motor homunculus can be represented. You'll notice that each portion of the precentral gyrus is devoted to a particular part of the body. As a generality, there's a greater amount of the motor cortex that's devoted to areas which have greater muscle complexity. So you'll notice that the hand occupies an outsized portion of the precentral gyrus, as does the face itself. So you'll see areas devoted to the lips, the jaw, the face, neck, brow, eye, areas that are involved in things like nonverbal communication, the tongue, swallowing, and so these are things that are involved in rather complex motions that require a lot of coordination. These are both motor maps and sensory maps, the motor maps in the precentral gyrus and the sensory map in the postcentral gyrus. And again, you'll see greater areas of the motor map of the motor cortex devoted to areas that have a higher degree of complexity in movement. When it comes to the sensory map, and we'll discuss this in more detail in just a minute, you see a higher proportion of the sensory map devoted to areas that have basically more nerve endings. While the motor cortex directly directs movement, the premotor cortex is basically what helps us plan movements. So essentially, it's a staging area for skilled motor activities. So it's a planning area as much as anything else. And so basically, this controls learned behavior or learned movements, repetitious movements, pattern motor skills. So this is where practice actually makes perfect. Typing, playing piano, using a fork, turning a page. And so essentially, your premotor cortex is going to enable you to carry out learned and repetitious behaviors without thinking about them quite as much. What the premotor cortex will also do is coordinate simultaneous and sequential actions of muscle groups. So it'll coordinate muscle groups that need to work together and muscle groups that need to work after one another. And this will also control voluntary actions that depend on sensory feedback. So if your movements are based on getting some sort of feedback from your muscles, from your joints, from your tendons, the premotor cortex will help control these. The other two motor areas which we saw or Broca's area, which is present in only one hemisphere, usually the left. This is a speech area that directs the muscles of speech coordination. So this essentially translates thought to speech. So Broca's area is active in planning speech, and it's also active in voluntary motor activities. The frontal eye field will control voluntary eye movements. So if you make tracking movements of objects within your eye field, and you're following them along with your eyes, that's what the frontal eye field is responsible for. So again, here we see the portions of the motor areas: the primary motor cortex, the premotor cortex, the frontal eye field, and Broca's area. Next, we're going to cover the sensory areas of the cerebral cortex. These give us conscious awareness of sensation. So basically anything that you feel, you're perceiving in these areas of the cerebral cortex. Now, the sensory areas are going to occur in all of the lobes except for the frontal lobe. So the parietal, insular, temporal, and occipital lobes. You may have noticed the frontal lobe is concerned with motor movements and other functions, which we'll cover in just a little while. Contained in these areas are the primary somatosensory cortex, the somatosensory association cortex, visual areas, auditory areas, the vestibular cortex, which is devoted to balance, the olfactory cortex, which is devoted to smell, the gustatory cortex, which is devoted to taste, and the visceral sensory area, which will perceive sensations from your internal organs. Let's take a look at the location of the sensory areas and related association areas. First, you see the primary somatosensory cortex and the somatosensory association cortex. These are involved in somatic sensation, which is basically the sensation of the position and movement of your limbs, so your muscles, joints, and tendons. Please notice that the primary somatosensory cortex is located in the postcentral gyrus. The gustatory cortex is located in the insular lobe. So if you look where the pointer is, it would actually be deep to where the pointer is. And again, this is involved in taste. The primary visual cortex and the visual association area are located in the occipital lobe, which may seem a little bit odd. This means that a blow to the back of the head could make you functionally blind if it disabled your primary visual cortex, despite the fact that you would still have fully functional eyes and optic nerve. Located in the temporal lobe, you find the auditory association area and the primary auditory cortex. Again, note that the primary somatosensory cortex is located in the postcentral gyrus of the parietal lobe. And again, what this does is to receive sensory information from skin and position or proprioceptors of skeletal muscle, joints, and tendons. So basically, what the somatosensory cortex can do is it can identify a region of the body that's being stimulated, and it can also basically inform you where your limbs are. If you close your eyes, you'll note that you can touch the tip of your nose without opening your eyes. You have an inherent sense of where your skeletal muscles, your joints, and your tendons are located. And so this is the sense called proprioception. And as we've seen before, a somatosensory homunculus, this represents contralateral sensory input from body regions. And as I mentioned before, there are larger areas of the somatosensory cortex devoted to areas with higher concentrations of nerve endings. So the somatosensory association cortex is posterior to the primary somatosensory cortex, and this is an area of interpretation. Basically, it will take input from the primary somatosensory cortex and interpret it so that you can determine size, things like size, and texture, spatial relationships of parts of objects that are being felt. The primary visual cortex, also called the striate cortex, is at the very back tip of the occipital lobe. And this does receive visual information from the retinas, which again we saw in the last dissection in lab. The visual association area surrounds the primary visual cortex. And essentially what this does is to remember past visual experiences to interpret what you're seeing now. So things like color, form, movement, ability to recognize objects. This comes from the visual association area. And this involves the entire posterior half of the cerebral hemispheres. So as I mentioned in lab, we are organisms that are very dependent on our sense of sight, and therefore we have quite a bit of our cerebral cortex devoted to the visual association area. The primary auditory cortex is located at the superior margin of the temporal lobes. And what this does is to receive information like pitch, loudness, and location. The auditory association area does basically the same thing for the auditory cortex that the visual association does for the visual cortex. It's located posteriorly to the primary auditory cortex, and it'll store memories not of sight this time, but of sounds, and it allows you perception of sound stimulus. The vestibular cortex, which wasn't pictured on the diagram, is located at the posterior part of the insula and part of the neighboring parietal cortex. And basically, again, what the vestibular cortex is for is conscious awareness of balance, which is pretty much the position of your head somewhere in space. The olfactory cortex is at the medial portion of the temporal lobes, and this is the region where we have awareness of odors. The gustatory cortex is in the insula. It's just deep to the temporal lobe, and this is, again, involved in perception of taste. The visceral sensory area is posterior to the gustatory cortex, and again, this gives the perceptions of visceral sensations, so upset stomach, full bladder, the stretch of our lungs. These are things that the visceral sensory area allows us to perceive. Sorry for the repetition here. We'll move on. Multimodal association areas. These are things that allow us to draw together inputs from multiple sensory areas. And these also send outputs to multiple areas. So essentially what these are going to do is give us meaning to multiple information. So if you needed to identify a dog, there's a certain shape and a certain sound that a dog might make, a certain odor that a dog might have. And so a multimodal association area will receive information from multiple association areas. And what this does is this allows us to make meaning of information that's received. It also allows us to store sort of this multiple input in memory. It gives us a connection with previous experience, which allows us to make better decisions in terms of what we're going to do about what we're sensing. And so multimodal association areas give us sensation, thoughts. This is where emotions become conscious. And basically, this is kind of what makes us who we actually are. There are three broad parts to the multimodal association areas. An anterior association area, which is called the prefrontal cortex, a posterior association area, and a limbic association area. So here we'll see the prefrontal cortex, which is in front of the frontal cortex, which is at the front of the frontal lobe, the anterior portion of the frontal lobe. And there are three different areas here. One has working memory for spatial tasks. So basically what this is going to do is it's going to allow you a memory of particular motions. An executive area for task management. So basically, if there's an order in which you're going to carry out a particular task, this is where it gets processed. And a working memory for object recall tasks. So if you're recalling something that you've done before, if you're carrying out a motion that you've done before, this is where you're going to do some of the processing there. Additionally, the very anterior portion of the prefrontal cortex, this is where we solve complex multitask problems. So mathematics, where you interpret things that require higher order thinking. This is what you're going to find processed at the prefrontal cortex. Finally, if you look at the back of the area, or at the back of the diagram, you'll see a second dashed area called Wernicke's area. And basically what Wernicke's area is going to do is it's going to do the opposite of what Broca's area does. So basically Wernicke's area allows you to hear particular sounds or to hear speech and interpret it. So again, the prefrontal cortex is involved with intellect, cognition, recall, personality. It is your most complicated cortical region, and it contains working memory for abstract ideas, judgment, reasoning, persistence, planning. So basically a lot of the abstract things that make up how we view the world. And the development of the prefrontal cortex depends on feedback from the social environment. So this is one of the reasons why it's absolutely vital that very young children have a lot of interaction with other people. The posterior association area, what this does is to play a role in recognizing patterns and faces, but it's also involved in understanding both the written and spoken language. And again, this is Wernicke's area. And this is located in the temporal parietal and in the occipital lobes. The limbic association area is part of the limbic system. And so this is going to involve the cingulate gyrus, the parahippocampal gyrus, and the hippocampus. So a couple of gyri, which we'll see in just a moment. And basically what this does is to inform what we're perceiving with emotions. So this is basically what infuses emotions into what we perceive. And so this makes what we're looking at important in some fashion, and it also helps firmly establish memories. I think you probably already know that some of your most intense memories are associated with intense emotional experiences. So the things that you remember the most or the most easily are the things that have meant the most to you in terms of emotional impact. So here you see the parahippocampal gyrus to the lower right and the cingulate gyrus. I mentioned lateralization earlier. First thing to tell you is that the hemispheres are almost identical, but there is some division of labor between hemispheres. And what we see with handedness is when a particular hemisphere is dominant for language. And so the left hemisphere is dominant for about 90% of people. And this also influences things like handedness. The left hemisphere controls language, math, and logic, and the right hemisphere controls visual spatial skills, intuition, emotion, and artistic and emotional skills. There are some people who will tell you that people who are right-handed have a better grasp of language, math, and logic. And people who are left-handed, in other words, right hemisphere dominant, have better visual and spatial skills, better intuition, feel more deeply, are better artists, better musicians. That's not something that I really buy into. I think as organisms, we're flexible enough to where someone who is left hemisphere dominant can be just as good an artist or a musician as someone who is right hemisphere dominant. And the reason why is because the hemispheres, they communicate almost instantly with tracks that lead in between them and what's called functional integration. In other words, if you're carrying out some sort of movement like walking or running, your hemispheres are going to have to coordinate. So there's going to be functional integration to carry out more complex movements. So there's a lot of communication between hemispheres. So that actually covers the gray matter that we've been talking about, at least for now, the cerebral cortex portion. Now we're going to go on and talk about what happens with cerebral white matter. And so white matter consists of myelinated fibers and tracts. And so again, remember that tracts are bundles of axons. These are the portions that are myelinated. And as we saw in the nervous system physiology chapter, myelination enables faster travel of nerve impulses. And so when you see white matter, you're going to be basically looking at an area that involves communication. And in this case, cerebral white matter involves communication between cerebral areas and between the cerebral cortex and the lower central nervous system. And by lower central nervous system, I mean the cerebellum, the diencephalon, and the brainstem. So association fibers run horizontally between left and right. I'm sorry, not between left and right. They connect different parts of the same hemisphere. So they would basically run anterior to posterior. Commissural fibers are horizontal, but in this case, they connect the gray matter of two hemispheres. You may remember the corpus callosum from your brain dissection. That's actually the biggest bundle of commissural fibers. Projection fibers don't run horizontally, they run vertically. So these run superiorly to inferiorly. And again, they're going to connect hemispheres with the lower brain or with the spinal cord. So here you can see that the association fibers are connecting different portions within the same hemisphere. I want you to notice that the commissural fibers that are in green, they're present just as dots. And what that means is that you're looking at a cross section of these fibers, meaning that they're running towards and away from you. And given how this brain is split, which is mid-sagittally, that is something that should make sense to you. The projection fibers are running from higher to lower portions of the brain. And so basically there are two major divisions of this, the corona radiata and what's called the internal capsule. And again, as I mentioned before, the main portion of commissural fibers is called the corpus callosum. The next thing that we're going to talk about are the basal nuclei, sometimes called the basal ganglia. Now, this is really unfortunate that some of them are called the basal ganglia, because one thing that I've told you in a prior lecture is that nuclei is a name for a collection of cell bodies within the central nervous system, and that ganglia this is a name for a collection of cell bodies in the peripheral nervous system. Yet, some groups of cell bodies within the central nervous system occasionally are called ganglia. This is really unfortunate, but this is kind of an exception to our rule. For the test, what I want you to remember is that ganglia is the term for a collection of cell bodies in the peripheral nervous system, nuclei for the central nervous system. So now let's talk about the subcortical nuclei. These involve the caudate nucleus, the putamen, and the globus pallidus. The combination of the caudate nucleus and the putamen is called the striatum. The basal ganglia are associated with what are called subthalamic nuclei located in the diencephalon and the substantia nigra, which are located in the midbrain. So there's some communication between the subcortical nuclei and the subthalamic nuclei and the substantia nigra. So here, if you take a look at this, you'll see the location on the left of the basal nuclei, the caudate, the putamen, the globus pallidus. And again, what these are going to communicate with are these other nuclei which are located in different portions of the brain. Again, for instance, the substantia nigra. Okay, this is a picture of the caudate nucleus and the putamen, which together again form the striatum. Again, another picture of the putamen and the globus pallidus. Sorry for the mispronunciation. There you also see the tail of the caudate nucleus. So what are the functions of these basal nuclei? Well, they're thought to influence muscle movements. They're also thought to play a role in cognition and emotion. They may regulate the intensity of slow movements or stereo movements, things like arm swinging. They may also filter out incorrect or inappropriate responses. So if you know somebody who doesn't have much of a filter with what they say, their basal nuclei may not work quite as well as somebody else's. And they're also going to inhibit antagonistic or unnecessary movements. So in other words, if you're trying to pick something up, you're going to want to have contraction of specific muscles and inhibition of other muscles that oppose them. And so basically, what the basal nuclei can do is to help coordinate movements such that you inhibit antagonistic motions that would stop the type of movement that you're planning on making. So this is the entire lecture in one splash right here. Cortical gray matter, it localizes and interprets sensory inputs. It controls voluntary and skeletal muscle activity, functions in intellectual and emotional processing. The basal nuclei are in charge of subcortical motor centers, which help control skeletal muscle movements. And that is it, I believe, for this particular lecture.